Yes. And apologies, first of all, for us.
Kicking off, yeah.
We had a few room issues here where the room we had booked was already in progress with the lecture, so we've had to hurriedly rearrange rooms, and so yeah, apologies for the late start. But we're very lucky today to have Dr. Joel Vanest from Plant and Food Research as our speaker. Joel's been working on kiwifruit disease. He's a bacteriologist, a plant bacteriologist, who's been working on kiwifruit disease and specifically PSA for many years now. He's a very distinguished scientist and won the Prime Minister's Prize for Science a few years ago. Yeah, and so our team and the seaweed team has been doing some work looking at seaweed extracts and how we can help fight some of these diseases with Joel and his work as well, and integrating the two together. So we're really lucky to have Joel today. I will pass it over to him. Welcome.
Okay, well, thank you very much, Rebecca. Thanks for the introduction, and thanks also for the invitation to come here to Tauranga to talk about something close to my heart, the syringae actinidiae, PSA, which is a bacteria on which we've been working for many years. So, and great also to have the opportunity to come here to Tauranga. So, because of course I work in Hamilton. And what happens when you come from Hamilton to Tauranga, you drive over the Kaimai, and when you arrive in Bay of Plenty, in the heart of the kiwifruit industry, what you see this, no, oh, it's not working. Sorry, I'll have to stay here. You'll see it's not working either. So this is working. No, it's not working. Sorry. Yes. Okay, so I'll have to stay here with the mouse. That's all right. Okay, so should work. Okay. Oh yes. Thank you very much. Yeah. So when you drive over the Kaimai and you arrive in Bay of Plenty, you see that sign. And if you know nothing about PSA, it tells you PSA, whatever it is, it's scary. It is frightening. It's something that's frightening the industry. And then you look at the sign and you see all the moss and all the lichens. And you say, well, clearly people don't care about it anymore. It's over. It tells you, well, PSA, whatever it was, has been solved. It was yesterday's problem. And what I'd like to do in the seminar is to see with you whether PSA actually has been solved. Is it actually yesterday's story? So what is PSA? Well, PSA is a bacterium that was first found in Japan in 1989, then in Korea in 1992, and Italy in 1992. It caused a number of symptoms on kiwifruit, and the symptoms are quite varied. You have cankers with production of a red exudate. You have discoloration of the tissue underneath the bark. You have production of exudate, which is that drop here, you see. So this is bacteria, pure bacterial culture coming out of the tissue because the tissue is so full of the bacteria. You find that also on tissue which do not show any of the symptoms. You have here a drop of exudate on some flowers. You have birds that do not develop in spring. You have canes that die, and ultimately you lose your orchards. All those pictures were taken in Italy in 2009. Before 2009, you would not have found any of the symptoms in kiwifruit in Italy, even though the pathogen had been found there in 1992. And the question is, why between 1992 and 2009, we didn't see any of the symptoms in kiwifruit orchard in Italy? So clearly the environment was the same. It was Italy, Latina, south of Rome. The genetic component from the plant, sorry, it was the same genotypes. It is cultivar they were growing before. So could it be that the pathogen had evolved, that the pathogen somehow was slightly different? So we went to Italy and got some of the pathogen, and we compared them with PSA that had been isolated from Japan and Korea, or Italy in 1992. To characterize those strains, we did what's called the box PCR, when you have one primer that binds a different area around your chromosome. By PCR, you'll have a number of amplicon on different sizes. And when you run them on the gel, you'll have a profile. and that profile will be characteristic of a strain or a group of strains. And doing so, we found that all the strains of PSA from Italy after 2009 shared the same profile, and that profile was different from the one from Japan and Korea. When we sequence few genes that are used in Pseudomonas syringae for taxonomy purposes, and that is in particular the CTS genes, we find that all the strains from Japan and Korea had the same sequence for that CTS genes, and all the strains from Italy had a different sequence, few differences. So we call those strains CTS haplotype A. A was for Asia, clearly, and the other one, CTS haplotype I for Italy. And it is really the strains from CTS haplotype I that caused all that destruction in Italy, all that destruction in 2009. However, all that started earlier. It started basically in 2008, when the only visible sign of an upcoming outbreak was few leaf spots and few necrosed flowers. So now, in November 2010, when in New Zealand we found few leaf spots and few necrosed flowers, it was whoa, and we knew it was PSA. The question was then, do we have the haplotype they have in Asia, which might not be such a big problem, or do we have the haplotype they had in Italy? So we went and collected some strains and did the box PCR as we did before. So we have here the profile for the strains from Asia, here the profile from the strains from Italy, and all the strains here were from New Zealand, and they can be in two groups. One group here has the same profile as the strains from Italy, and that was bad news. But then we had that group of strains here that had a profile which is different from the one from Italy and the one from Asia. And we knew that all those strains were actually PSA based on the PCR treatment we developed, the primers we developed before. So as far as we knew, this was all PSA. Now, we knew that those strains here would destroy your orchard, and inoculated on young plants in the laboratory would cause a lot of damage. On the other hand, those strains here were found in an orchard where we didn't find any other symptoms. And when you inoculate them in plants, you barely could see any leaf spots. So to differentiate those two groups, we call this group PSA-V for virulence and PSA-LV for low virulence. Those strains here, LV, seem to be, had been found at that time only in New Zealand. So we were very interested to know what are those strains. So we did a little bit more work on them. And doing more work, we find out that actually genetically they were different enough from PSA. And the pathogenicity was different enough from PSA that actually they qualified to be a different pathovar. And now they are called Pseudomonas syringae pathovar actinidiae. So PSA-LV is actually not PSA. PSA-V is PSA. It is actually one of the 5 biovars of PSA. Because in PSA, you can differentiate based on the genetics and other characteristics. You can distinguish 5 different biovars. Biovar 1, 2, 3, 5, and 6. Biovar 4 became PFM. Pseudomonas syringae actinidiae. And all the strains we have in, all the strains we have in New Zealand belong to biovar 3. And that's the biovar that has been found also in Italy, has been in France, has been found in Chile, has been found in China. It is the biovar that's causing a lot of trouble to the kiwifruit industry around the world. However, in New Zealand, I think PSA has spread faster through the industry than in other countries. And here on this slide, we have Bay of Plenty. All the little dots of different colors represent kiwifruit orchards. In red, you have the orchard of the area affected December 2010. That's when we discovered the beginning of the outbreak. And within 1 year, by December 2011, most of Bay of Plenty was actually infected by PSA. Within few years, if you look at New Zealand, and every dot represents kiwifruit orchards. Only around Whangarei and the north of South Island, that those two areas were only areas that were PSA fruits. So why PSA spread so rapidly in New Zealand? Well, a number of reasons. The first reason is actually environmental conditions very favorable for the organisms. If you look here, you have the maximum temperature and minimum temperature in Tauranga. And if you compare with Latina, where they grow kiwifruit in Italy, or Matsuyama in Japan, you see they have very high, extreme high temperature and extreme low, which we do not have in New Zealand, not in Tauranga. Which means year-round, the temperature allows the bacteria to multiply, allows the bacteria to establish and to infect. So we have an organisms here that can infect year-round. The other reasons why it went very rapidly is that you have this continuous kiwifruit after kiwifruit. So once the pathogens in there, it finds a fantastic terrain to move. And of course, all that was with a narrow genetic base because all those orchards are basically two cultivars, Hot 16A, the yellow-fleshed cultivar, and Hayward, the green-fleshed cultivar. And this yellow-fleshed cultivar, Hot 16A, is very susceptible to pests. Actually, it is the one that took the brunt of the outbreak. If you look here so that is actually the area of Hot 16A that was grown. And you can see after PSA came in, it went down. And that, of course, the production of Hot 16A follows the same curve, the revenue, and so all follows the same. And you had all those headlines, the paper, 100 million disaster, gold kiwifruit exports halved. This was actually the dotted line, excuse me, that dotted line here was actually the prediction of how Hot 16A and yellow-fleshed kiwifruit was going to do in New Zealand, and you can see it was going to go up and up and up and up, except that PSA came in and ruined the show. Now we do know that was in 2010 or 11. Now we know that it is not quite the case. We know, and if we follow the papers, that things have changed. In this headlines here, you have green turns to gold for Zespri. The kiwifruit industry has come back stronger following the ravages of PSA disease. I've seen presentations, so that was a few years after the beginning of the outbreak, where people were super optimistic, and you had the slides out of the darkness. Thank you to all who contributed to this miraculous recovery. Wow, those are headlines. So let's look a little bit at how we came to that miraculous recovery. As soon as PSA came in New Zealand, Zespri asked the question, and that was, I think, an interesting question, an important question, is there anything out there that exists on the shelf we could take, we could use to reduce disease incidence? Anything at all? Yes, and that was important to do. And so we did it, and we did over 300 treatments, and we looked for either direct inhibition of the pathogen, or elicitation, means a process that makes the plant more resistant to the pathogen. Over 30 treatments. Unfortunately, at the end, we didn't find many compounds that we didn't think would be good at controlling plant pathogenic bacteria. What we found antibiotic, streptomycin, casugomycin are very good at controlling bacteria, not a great surprise. We found that copper worked well, not so well in glasshouse experiments, by the way, but in the field worked very well in controlling PSA. And we found that Bion, or ASM, acetylbenzoate S-methyl, which is sold as Actigard, some part of the world as Bion, which is an elicitor, also reduce disease incidence. We looked at biological control agents, and although some of them were working, in general, the resistance or the control was not consistent enough for us to make recommendations about those back control agents. And it is not very surprising because those back control agents were selected for the ability to control different pathogens on different crops in different environments. But of course, since then we know, Phil Elmer and Stephen Holt developed this this Oreo gold, which is a biological control agent, which is a yeast-like organism, Oreobasidium pullulans. And so all those products are now available for people to control PSA. And that represents, I would say, one of the pillars of this miraculous recovery. The other pillar is actually the plant by itself. So Stephen Hoyt spent a lot of time phenotyping cultivars of kiwifruit. Actinidiae chinensis, the yellow flesh kiwifruit, Actinidiae chinensis deliciosa, the green flesh kiwifruit, and a couple of others. And he developed a wood stem bioassay, and he could rank all the cultivars against that bioassay index. In the same time, people work in the field developed a field score, and you could rank all the cultivars by that field score. What is clear is that Hot 16A, the yellow flesh cultivar we had before PSA came in, is very susceptible, falls over really quickly in the field, and also with the stem bioassay. While Gold 3, a new cultivar that was in pre-commercial trials when PSA came in, and the yellow flesh kiwifruit turned out to be much more resistant in the field and also in the root bioassay. And that Gold 3 represents the second pillar, I would say, of that recovery that allowed the industry to keep going. So, and actually, there's a third pillar to that recovery, and that has to be the hygiene. If you want to control PSA in your orchard, you really need to have hygiene, is removing the symptom, removing the inoculant. And with three pillars, when they put together, allows people to grow kiwi fruit and not to worry so much about PSA as long as they follow the rules. Now, you need to put all that together, and this is something that has been put together by KVH, Kiwi Fruit Vine Health. So on the top, you have here a whole season of kiwi fruit going from bud break to dormancy. And here all the product and when you can use them. So all the product available in New Zealand today are all those products, which are copper-based products, and you can use them most of the year except during flowering. Then you have bactericide, casugomycin, and streptomycin. You can use only for a very short period of time. And then you have Oreo Gold, which is called biobactericide, which I think is a fancy name for biological control agent, which is the same as botryzane and Kiwivax. And you have two compounds, Actigard and Ambitious, which are two compounds which do not affect the pathogen, but affect the plant and make the plant more resistant to infection. And so following all those, using all those compounds and hygiene and Gold 3 allowed us to move from this picture to this picture. So this picture here, you have the decline of Hot 16A following the outbreak of PSA. And you see that soon after Gold 3, which is the name of the cultivar ZC002, grew and in few years represented all the yellow-fleshed kiwi fruit produced in New Zealand. And to a point where, in actual fact, if you look here, it is a level of production or area being planted, which is what Zespri would have predicted or wanted to be at before PSA came in. So if you look at it, it's a bit like PSA, which is represented by this pink area here. In hindsight, you can look at PSA and say, well, it was like a bump in the road, so to speak. You know, it slowed down where we wanted to be with yellow-fleshed kiwi fruit. It didn't stop it. It's a scary bump in the road because, you know, we didn't know whether we would get it out of it. And very expensive as well, 90 million trays equivalent. That's a lot of money, right? But in hindsight, it looks like, well, it's behind. And so this explains now that slide with the toolkit, which I just talked about, and that Sun Gold, which is how the fruit from Gold 3 or ZC02 are marketed. So has PSA been solved? Is it just the storage? And I have to say, if I were a grower, I look at those graphs, I look at all the products available, and I looked at all the problems I face, which is low chill in winter, means not enough flowers in spring, late frost in spring, not enough people to pick my crop, not enough people to prune my crop, or to prune my orchard, and then the consequences of climate change, flooding that occurred here in Bay of Plenty. And I said, well, I have plenty of things that seem to me to come ahead of PSA. And I'm tempted to say, yeah, well, PSA is not right there on my radar. Yes, and I understand that. As a grower, I understand that. Now, as a scientist, well, I have a different story. I'm not convinced that it is solved. I'm not convinced it is yesterday's story. Actually, I'm convinced we should not be complacent. And with the CEO of Intel that said, success brings complacency, complacency brings failure, only the paranoid survive. So we'll jump in it, and I'll tell you why I think it's probably important not to be complacent. Let's look at the first tool we have today to control PSA, which is copper, all those copper products. They were used from 2011. By 2015, two teams in New Zealand, Elena Colombi and Ona McCann in Massey University, Russell Poulter and Maggie Butler in Otago University, independently found some strains of PSA that were resistant to copper. 4 years after we started to use it, not a huge amount of time. So we were interested, can we still control PSA with those strains with copper? So we went into the glass house and we did the experiment, and we have here 10627, which is a strain which is copper sensitive, and 3 strains that are copper resistant, and those are resistant to the highest level of copper. So this is the strain that was the most resistant strain we could find in New Zealand. And in the glass house, yes, we could control all those strains. We were very happy. But as I said, copper doesn't work that well in glass house. So then we went into the orchard and we used some trap plants, and we asked the same question. Can we control PSA, copper-resistant strains of PSA using copper? And yes, we can. If you look at the number of leaf spots on plants that were treated with water versus copper, there's a significant difference. But here it is where the good news stops. Because from the trap plant, we analyze each leaf spot, and we ask the question, is this leaf spot caused by a strain which is copper resistant or a strain which is copper sensitive? And the result is that this is the ratio copper resistant, copper sensitive we had in our water treated plant and in our copper treated plants. So yes, we are controlling the strains, but we're increasing the ratio copper resistance. And why is that a problem, you will say? Well, it is a problem because we have here on this graph, this schema, you have the concept of cupric ion. Cupric ion is what kills your bacteria. So the copper product come in different formulation, but it's the cupric ion. And that cupric ion is released over time. And so you have the concentration, and over time it goes down. There is a level of copper under which there is absolutely no effect. In fact, the bacteria is quite happy because the bacteria needs a bit of copper to survive. So if in your orchard you have copper sensitive strains, you kill your bacteria from that point to that point. So during all that time, your product is effective. If now you have a mixture of copper resistant and copper sensitive strain, and let's say for argument's sake that the level of resistance is represented here by this yellow line. When the concentration of cupric ion goes beyond that yellow line, you control only the copper sensitive. So you increase the copper resistance to the point where there will be no more copper sensitive, in which case you control PSA only in that shorter period of time here. So you still control, but your product is effective for less longer, for a shorter period of time. And if you think, and at the time that was the thinking, I'll be smart, I'm going to use lower concentration of copper, but many times. If you do that, well, this is what could happen. You would have a loss, total or partial loss of control until you cross that line. And then, and only for that short period of time, you'd have full control of PSA. Unfortunately, it gets even worse because both teams looked at copper resistance, found out that the genes for copper resistance, so copper resistant, the bacteria is copper resistant if it can capture the copper and eject it or trap it and store it somewhere. Okay, all that requires genes that produce enzyme and protein to do that job. So those genes have to be acquired. And the genes, it's classic among bacteria, copper resistance genes are very often, if not all the time, carried by plasmid, which is a small element here that can move from bacteria to bacteria. Those two bacteria don't have to be even the same species or the same genus. They don't need to multiply. And they also found that those copper resistance were carried by what's called the ICE, integrative conjugative elements. You have an element here. So that strain, let's say, would be copper resistant, and this one is not. And in no time, both of them are copper resistant. That explains why copper resistance can spread through a population very rapidly without the bacteria multiplying. It can spread from one species to another, horizontal gene transfer. Now you'll say, well, it can transfer, but we still control it. So where's the problem? Well, here's the problem. This is a number of strains we isolated in New Zealand, all from New Zealand, all from orchards, not kiwifruit orchards, but different orchards. And you look at the level of resistance of the Pseudomonas syringae. If you look at Pseudomonas syringae actinidiae, PSA, this is this strain which is resistant to the highest concentration of copper. Concentration of copper is here. In that assay, these strains can resist to 1.25 millimolar. But in other orchards, summer fruit orchard, pit fruit orchards, we found strains here that resist to 2 millimolar copper in that same experiment. Most probably those genes are also carried by mobile genetic elements, plasmid, ICE, that could move horizontally. And the question is, what would happen if those elements here would move into PSA? Could we still control those strains with the level of copper you're using today in the orchards? And we don't know the answer to this. So far, we haven't found, and KVH is looking at it every year, we haven't found a strains of PSA which is resistant to a higher concentration of copper than this one. There is perhaps a very good reason for it. Biological reasons, but I don't know it. Nobody knows it. And so therefore, at this stage, we cannot rule out that one day we'll have strains that resist to a higher concentration of copper, which means we won't be able to control them with copper. And it gets worse. So this is the genetic organization of copper, of the genes involved in copper resistance. And so they're all copper resistant, CUS, and so on and so forth. But embedded in them, you have those two genes, STRA, STRB. Those genes will give resistance to streptomycin. And in this case, they're embedded in the middle of the copper resistance genes, but in other cases, they are on the same mobile element, a little bit further, but same mobile elements. So that means when a strain receives one of those mobile elements, if you use copper, you give a selective advantage to the strains with the copper resistance genes. At the same time, you give an advantage to the strain which are streptomycin resistant. You have a resistance. And so you cannot use streptomycin later because your strain is already resistant. So, well, you say, well, we still have the elicitors, don't we? Why don't we use elicitors and get that plant pumping and being resistant as much as you can? Well, this is work from Tony Redensky. And so you have a kiwifruit plant here that was not treated, and here one that was treated with ASM on a regular basis, week after week. And if you do that, what you find that your plant doesn't grow nearly as well as the plant that was not treated. There is a trade-off between resistance and growth. So you can't make your plant resistant all the time because it will stop growing. Okay, so that's one of the pillars that looks a little bit more shaky than we would like. If you want perhaps a good news about that pillar, is that about castigomycin resistance, so far we have never found a strain of PSA which is resistant to castigomycin. On the downside, we know that calcium resistance exists in New Zealand instead of Pseudomonas, but we do not know whether that resistance is carried by mobile genetic elements and therefore whether that resistance could be transferred to PSA. So now let's look at the second major pillar of the miracle, which is gold-3, which is resistant to PSA. So what happens when a bacteria comes in contact with a plant cell? The plant will sense the presence of the bacteria, and that will trigger a series of signals that tell the plant there's a bacteria would possibly in danger. Let's put some defense. It's what happened. And so that bacteria will not cause any disease. And in most cases, most bacteria on most plants do not cause disease. Disease is the exception, not the rule. So what does happen? What needs to happen for bacteria to be pathogen? Well, what happens is that the plant has developed a system where they inject into the plant cell some effectors protein, which disrupt either the sensing or the signal transduction or the response of the plant. In this case, the plant is not responding, and you have disease. So a bacteria can become pathogen by acquiring genes that cause for those effects. Of course, during evolution, the plant has developed a system to protect itself from those effectors, and the plant has those resistance protein here that will recognize presence of the effectors. And when the plant recognizes that or detect with an effector, it will start a very strong defense. And so in this case, you will not have disease. But what happens if your pathogens only here, which cannot cause disease, would lose the ability to produce the effectors that are recognized by the plant? Well, what will happen is very simple. The plant will not recognize with an effector. It will not produce that strong defense reaction, and it will be susceptible. And your bacteria is pathogen. So your bacteria can become pathogen either by acquiring genes for effector genes or by losing some effector genes. So that's a bit counterintuitive in the sense that you can gain a function by losing some genes. That's how it happens. And Matt Stapleton, Dujiangyan, and Lauren Emara, who are working a lot on evolution of PSA in New Zealand, found actually that this does happen. PSA is evolving in front of our eyes. And so Actinidiae aguta, which is the kiwi berry, is resistant to PSA. However, they were able to find some aguta with symptoms caused by PSA. They analyzed the PSA and they found there's a deletion of those effector genes. We put those plants in a glass house, and I don't know if it's here, but when we use the strain with the deletion, we had all the symptoms, or if you put the wild type strains, new symptoms. Then those gels represent simply the fragment, and you get the fragment that's missing. So now those strains haven't taken over, and aguta is still resistant to PSA because in addition to being able to cause disease, they have other problems in virulence and multiplication, so on and so forth. But what I think this signals is that if we cannot take for granted that aguta is resistant to PSA, why are we taking for granted that gold-3 is resistant to PSA? We do not know the mechanisms of resistance. Could it be that a simple deletion would lead to gold-3 becoming susceptible? Okay, so that's fine to talk about all the difficulties and all the problems with the miracle, but the question is, what are we doing about it? It's nice to say, well, yeah, very good question. What are we doing about it? Well, actually, a lot of work is happening, and I can't talk about all the work. But I'll talk about some of it, and in particular work doing in collaboration with Waikato University, being here on the Waikato campus. I think it's probably the best thing to do. And so we had a collaboration that started many years ago with Michel Princep, Chris Butler-Hills, and Louise Dean, with a PhD student at Waikato. And the idea there was, is there some compound in seaweeds from Bay of Plenty that could inhibit PSA? So was a screening that was made of a number of seaweeds. Extracts were made, they were characterized, we checked in vitro for inhibition of PSA, and we found a number of them that do indeed inhibit PSA. On that slide here, you have, so this is the underside and the top side of the same leaf. On this area here, we had PSA by itself, and you see the necrosis happening. And here we had PSA with some of the seaweed extracts, and absolutely no symptoms. So very happy. Another collaboration, another system. This is with you, Mary Mason and Chris Glasson, where there we're looking at something different. We're looking at polysaccharide from all the seaweeds, and we're not looking at inhibition of PSA. We're looking at whether some of those compounds could elicit a reaction in the plant and make the plant more resistant. This is the very beginning of the project, but at the very beginning, there's already some compounds here that, when sprayed before PSA, reduce disease incidence. The great thing about elicitors from seaweeds and polysaccharide is that it would give now the organic kiwifruit growers an elicitor, because Bayan or Actigard cannot be used on organic orchards. And we know that the organic sector is growing fast. And so therefore, I believe there is a demand for organic elicitors. And so that would be that could become the answer. What else? Well, so all that, if you think of it, what we're doing here, and there's another project with the John Innes Institute, where we're looking at biological control agents. And when we do there, we add more tools to the toolbox. That's what we're doing. In addition to that, we're looking also at novel ways of controlling PSA. And it is where Chuan Chigwo, who is right here, comes in. This is Chuan. You recognize that the laboratory in Chengdu, in Sichuan, yes. And so Zhuang, who is actually the co-director of the Kiwifruit Research Institute in the joint laboratory in Chengdu, is from Sichuan. So Sichuan is that central part here in China. Now, we know that kiwifruit is not from New Zealand. It is actually from China. And it is actually a lot of the kiwifruit are coming from this area here in China, which is where initially we got the first kiwifruit. Something that struck me when I went to visit Zhuang in Sichuan is that if you look at the orchard, they're hammered by PSA. This is a Hongyang orchard completely devastated by PSA. All around that orchard, you have the mountains and the wild forest where the kiwifruit is growing. You go in that forest and you will not find PSA. Zhuang went in that forest, looked for it, came back with thousands of strains, not a single one was PSA. On a McCann one with other scientists from Northwestern University, spent 6 months in the mountain, went throughout, not just Sichuan, Shanxi, Hubei, several provinces, came back empty-handed. And you have to wonder, why? What's happening? Why you have so much inoculum here and you have those diseases and nothing in the mountain? What's different? Well, clearly, it can't be just the climate. It can't be. So could it be that in actual fact, it is the microbial environment that would prevent PSA? And I'll tell you quickly how we came to that hypothesis. Years ago, I worked with Francesco Spinelli in Bologna University. We have a long relationship and collaboration that's going for years. And working together, we found that if you expose PSA to a supernatant in which you had a lot of PSA growing in there, so the bacteria was gone, but the secondary metabolites were left. If you do that, you find that some genes like OBZ5, which are involved in virulence, are overexpressed. And the genes involved in mobility are overexpressed. That tells you that PSA communicates with all the PSA cells. It's called quorum sensing. And when you have a lot of them together, they know it through chemical signals and they turn on the virulence genes. We did that experiment now with different organisms, syringae, Pseudomonas verdiflava, and fluorescence, which are bacterial species you find commonly on kiwi fruits. And when you do that, you find that again, PSA respond to the presence of those bacteria by turning on genes for mobility and turning on genes for biofilm formation, and also some genes involved in virulence. Oops, yeah, right there. So PSA communicates with its microbial environment and responds to it by turning on genes involved in virulence. Now, what would happen if we would break that communication? Well, there's a very good example of that. This is another bacterium. It's a Pectobacterium carotivorum that causes soft rot on potatoes. And when you let the communication occur and you put your Pectobacterium on a potato slice, you have all these rots. You prevent communication and you have nothing. So could it be that in the mountain in Sichuan and the rest of China, you have a macroflora that somehow modify the communication of PSA in such a way that PSA doesn't cause disease? To answer that question with Zhuang, we looked at the microbiome of kiwi fruit grown in Sichuan in an orchard with the kiwi fruit grown in the wild. And I'll show you just the very beginning of where we are. This is the very beginning of the analysis. It's just one site. Zhuang collected samples from 3 different sites. We're looking at bacterial microbiome, we're looking at fungal microbiome, we're looking at epiphyte microbiome, endophyte microbiome, and root-associated microbiome. So 6 microbiomes per plant. So a lot of work. This is just the very beginning. But I just want to show this because it is clear when you look at this taxonomic plot here that all the strains here, or all the microbiome from those plants here that's commercially grown, are very different from those that are in the wild. And one of the main difference is right here. It is, look at the diversity we have in the wild kiwi fruit that we lost in the commercially grown kiwi fruit. And we know that when we domesticate a plant, we lose diversity in the microbiome. And we know that diversity in the microbiome brings a lot of function to the plants. And one of the questions, and now, so we started this really looking at, could we explain the fact that PSA is in the environment in China and we don't find PSA in the wild kiwi fruits in China? That was the beginning of the story. But now when you look at this, what else has been modified in the short amount of time we domesticated kiwi fruit? The domestication of kiwi fruit might have resulted in a loss of diversity. What else? So there's, of course, resistance to PSA, but resistance to perhaps other pathogen, to pests, ability to deal with drought, ability to deal with flooding. What else has been lost? And once we find what has been lost, how can we bring it back? How can we use that microbiome to actually allow people to grow the best kiwi fruit in the condition in which they are? And so this is just the beginning of the study, but I thought it was very, very interesting. I'm very excited by it. Okay, so I think at the end of my time. So I just, before finishing, I want to thank Zespri and KVH who have funded most of the work I've talked about, and also Plant and Food Research through some CRIP programs. They helped some of the work. So thank you very much to Zespri, KVH, and Plant and Food. People in my lab who did the work, Deidre, Janet, Jenny, and Megan. PhD student that came in my lab and did some of the work I talked about today, Luca from Bologna, Amélie from France, Louis from Waikato University, and Javier Martinez from the John Innes Institute. People from Plant and Food Research, Tony, Kirstine, Steve, Matt, and Jida Rayman. People from Waikato University, let's not forget Waikato University, right? So Michelle, Chris, Mary, and Chris are here. People from Massey University, Onamakan, and then a lot of collaboration overseas, long-term collaboration, especially Francesco Spinelli, we collaborating for more than 20 years together. Jack Malone from the John Innes Institute, Marie-Agnès Jack from INRA in France, and Zhuang Hugo, who is here. We've been together since 2009, if I recall well. So, of course, at the end will be a disclaimer for the presentation, and otherwise I'll stop there. So thank you very much.
Well, what a fascinating seminar. We're really tight on time for questions. So I think we might just leave it just in the interest of time. This is just a small token from us to say thank you so much for coming over and for the presentation.
Thank you.
I'm sure if anyone's got any questions, they're welcome to contact you.
Yes, absolutely. There's my email there, and anybody with any question, I'd be very happy to reply directly to that. Yes.
Thank you so much, and thank you to everyone else for attending today.
All right. Thank you.
Joelle.
Mm-hmm.